So parts of the cell, starting with cell body, you can see there is a central nucleus, and within the nucleus, there is a prominent nucleolus. So this gives an appearance called owl eye appearance to the cell body. This is how you'll identify the neuron in histological slides. You will see a prominent nucleolus within the central nucleus. Now, the cytoplasm of the neuron is known as neuroplasm, and the cell membrane of the neuron is known as neurolemma. So in this diagram, you can see that there are a lot of cell organelles within the neuroplasm. So can you just type in the chat, what are the cell organelles that are usually seen within any animal cell? What are the cell organelles? Yes, there are the mitochondria, which is the powerhouse of the cell, the lysosomes, endoplasmic reticulum, Golgi apparatus. Yes, you're all right. You all know the cell organelles. So you can see within the neuron also, all these cell organelles are present. But there are certain specializations. For example, the rough endoplasmic reticulum within the neuroplasm. It is known as missile substance or missile bodies. You can see that labeled here, these small rounded structures, which are intensely basophilic. They're also called chromatin bodies or tigroid bodies. So this is an important feature that you have to know. The missile substance is present within the neuroplasm, the cell body of neuron. Now, there is one region within the cell body. I want you to focus on this area. It is called the axon hillock. Okay, so the entire cell body contains missile granules. You can see these rounded structures. But in the region of axon hillock, that is where the axon is taking origin. Where the axon hillock is present, missile body is absent. That is an important feature that you need to remember. The missile body is present throughout the cell body, except at the region of axon hillock. Now, apart from these organelles and missile bodies, there are also certain filaments within the cell body. These are called neurofibrils, and they stain with a silver staining. Other component within the cell body are pigment molecules. So here you can see, this is the cell body with a central nucleus and a prominent nucleolus. And you can also see a few basophilic dots, which is the missile substance. Apart from that, you can see here is a cluster of pigments, okay, the pigmented area within the neuron. So these pigments are present in certain specialized areas in the central nervous system. A few examples of the pigments are neuromelanin or lipofuscin. So have you heard of lipofuscin before? Okay, what is the peculiarity of the lipofuscin pigment? You can type it in the chat. Okay, I think you have not heard of lipofuscin before. It is called a wear and tear pigment, lipofuscin. Okay, you will learn it a little later in other regions of the body. So out of these pigments, learn at least 2 examples right now. And the site of these pigmented neurons, you will learn when we learn the anatomy of the nervous system in gross anatomy. You will also be seeing it in dissection, certain pigmented areas within the brain. Okay, so this is the first diagram that you need to learn to draw. Okay, so here we can see the nerve cell or the neuron. The stain that we use is called toluidine blue stain. So when you are drawing this diagram, you need to draw it with your blue color pencil, with your light blue color pencil. Don't use hematoxylin or eosin for drawing this diagram. So you can see this is the star-shaped cell body. And there is the central nucleus with a nucleolus. You're drawing the missile substance as basophilic oval structures. Missile body is absent in the region of axon hillock. And there are many multiple branched processes in the cell body called the dendrite, and a single long process called the axon. Okay, the region of the cell body where the axon originates is called axon hillock, and the starting point, the initial portion of the axon is called the initial segment. This is how you draw the diagram of the nerve cell. Now we will learn about the neurites or the cell processes. You can see these are the short branching dendrites, and this is a single long axon. So based on these arrows, can you tell me what is the direction of nerve impulse transmission within a nerve cell? You know that the function of the neuron is to transmit the electrical impulses within the nervous system as signals for the body. So what is the direction of transmission of these impulses within the processes? Do you have an idea of the function of the dendrites and axon? Yes. Dendrites will carry the impulse towards the cell body. Yes. And axon will carry the impulse away from the cell body. Okay, good. You know that basic fact. Then just a few extra points is that the dendrites will contain missile bodies as well. Okay, the spelling is NISSL alone. And the dendrites are branched and multiple. They are irregular as well, whereas the axon will be long and having a uniform thickness. Missile substance is absent. And in the entire length of the axon, there is no branching. Then you know about the origin of axon, it's axon hillock. The proximal portion of axon is called the initial segment. At the end of the axon, okay, I told you in the entire course of axon, there is no branching, but at the terminal portion of the axon, there are few branched filaments and there are small swellings at the end of the terminal branches. These swellings are called terminal button. So the structure of dendrites and axon is also something that you need to learn. Now this terminal button of axon, it will be in contact with processes of other neurons within the central nervous system. So this communication between the terminal button and processes of other neurons within the central nervous system is called a synapse. Yes, so synapse is another important area for you to learn. In physiology, you will be learning about the structure of the synapse, the diagram. Okay, you need to know the parts of the synapse and also the classification of synapse. Okay, so in central nervous system, there is this communication between neurons through synapses. Whereas in the peripheral nervous system, okay, in this diagram, you can see the terminal button in the peripheral nervous system, it ends in an end organ. If it is a sensory nerve, it will end, the terminal button will end in a sensory receptor. And if it is a motor neuron, it will end usually on the skeletal muscle to form a specialized structure called the motor end plate. I think these terms are familiar for you if you've learned it in physiology. Okay, so that is how the terminal button will end, either in synapse in the central nervous system, or in the peripheral nervous system, it will end either in a motor end plate or in a sensory receptor, the end organ. Types of synapses, you have to read this from your textbook. This is just to give you an overview. Axodendritic synapse means that the terminal buttons of axon will synapse with dendrites of adjacent neuron. Axosomatic synapse is when the terminal buttons of axon will synapse with the cell body of adjacent neuron. There is a synapse known as axonic. Okay? It's between axons of two neurons. And there's also dendrodendritic synapses. Suppose a short note is asked for you on synapse. You have to draw the diagram about parts of synapse and the types of synapses. This is another word for terminal button, a French word called bouton terminaux. So in this electron microscopy picture, this is actually an axosomatic synapse. So all of these smaller processes that we see, okay, single process, it's actually the axon of surrounding neurons. And this is the terminal button of the axon, whereas this is the cell body of a neuron. So I just want to show you the appearance of the terminal filament and the terminal button of axon. Whereas in this picture, okay, this whole green knob is the terminal button. And here is part of synaptic cleft when neurotransmitters are released into the synapse. Okay, these are the vesicles of neurotransmitters which are going to be released into the synaptic cleft. Okay, so you told me that the cells of myelination in the peripheral nervous system is the Schwann cell. So does anyone know what is the cell of myelination within the central nervous system? Yes, good. In central nervous system, it is oligodendrocyte. You just have to learn this. So now we're going to deal with the myelination by the Schwann cell. Okay, I think I'll just give you a 1-minute break here, okay, because we're going to learn a little in detail about myelination. Okay, so just refresh your mind for 1 minute. I'll give you a small break. Don't worry, I'll let you go for your longer break on time at 11 o'clock. Okay, class, so we are resuming. Can you just give me a confirmation of that you're back?
Yes, ma'am.
Okay, yes.
Yes, ma'am.
Okay, thank you.
Yes, ma'am.
Okay, so now let's look at the process of myelination. Okay, as we have learned, look at the parts of the cell here. There is the cell body. There are dendrites, multiple branched processes, and there is a single long axon. Now, look at the cell which is surrounding the axon, okay, or the cell which forms a sheath around the axon in the peripheral nervous system. This is the Schwann cell. So let's look at how the Schwann cell produces the myelin sheath. The process of myelination, it is something that you have to learn because it will be asked from an exam point of view. It is asked in your university exam. Okay, or even as a point of interest, you can learn it. Okay, so here is the Schwann cell with the nucleus, and this pink structure is the cytoplasm, whereas the yellow is the cell membrane of the Schwann cell. Look at the axon of a neuron, how it is indenting into the Schwann cell. And we can see that the ends of the Schwann cell, as indicated by arrows, the ends is going to get opposed to each other. Now, when the two ends of the Schwann cell get close to each other, there will be a fusion of the cell membrane of Schwann cell. So that forms a structure called mesoaxon. Just remember that the fused end of Schwann cell, the cell membranes will also get fused to form the mesoaxon. Then we can see this mesoaxon, it will elongate and it will spiralize. And the cell membrane of Schwann cell, it will secrete cholesterol and phospholipids. So that is what helps to form the thickened myelin sheath. So this yellow color cell membrane is what secretes the myelin to form the myelin sheath. Meanwhile, the cytoplasm of the Schwann cell will get thinned out and pushed to the periphery. So you can see the difference in the cytoplasm from when myelination was starting up to the completion of myelination. This process of myelination, it starts in utero, that is 4 months when the baby is in the womb, and it will be completed only at the age of 3 years of life after birth. Okay, so when you look at this picture, okay, this spiral of the myelin sheath, does it remind you of any other structure? Okay, the spiralization of myelin sheath, there's a special name for this process given by the scientists, okay, based on this appearance of myelin sheath. Does anyone know? Okay, think these scientists are kind of fun people, okay, or they are real foodies, okay, because they have compared the myelin sheath with this. Okay, so what is this snack? Okay, I think I'll just prepare your tummy for your break time soon. That's why I'm showing you these pictures. Okay, can anyone just identify what is this? Yes, jam roll. Okay, yes. The scientists have named the myelin sheath in comparison to this Swiss roll or jam roll, or they call it the jelly roll. Okay, because just like this jelly roll has spirals here, the myelin sheath is also formed by spiralization and deposition of myelin by the Schwann cell. So this whole process that we learned, all the steps that I've described so far, it is called the jelly roll hypothesis of myelination. So you can see in this electron microscopy picture how the myelin sheath indeed does have a jelly roll appearance. So this is the process that I have described. First, there is invagination of the axon into the Schwann cell, mesoaxon formation, then there's elongation and spiraling of the mesoaxon, and deposition of phospholipids, okay, there's proteins, and also cholesterol between adjacent layers of cell membrane of the Schwann cell. You can learn this process with the diagram. Now, what are the nodes of Ranvier? Okay, this is the whole myelin sheath. You can see that one Schwann cell will actually form one segment of myelin around the axon. So there are certain gaps in between two segments of the myelin sheath. the gaps which are present between the segments of myelin, that is called the node of Ranvier. So here in this H&E preparation, you look closely. Okay, so this is one segment of myelin. This is another segment. And here by the pointed arrow is the node of Ranvier. So again, see, this is the node of Ranvier between two segments of the myelin sheath. And I told you the cytoplasm of the Schwann cell will be pushed to the periphery. So between the two nodes, the segment of myelin is also called an internode. Now I want you to look at this structure, okay, which is a cytoplasmic tunnel or a cleft of cytoplasm, which will be entering into the myelin sheath. That is a special name called the Schmidt-Lantermann cleft. Okay, that is another important structure, especially if you're aiming for a distinction in your exam. You have to remember this name, okay, the Schmidt-Lantermann cleft. It's actually oblique cleft within the myelin sheath, and they are cytoplasmic tunnels. And it helps as a path for conduction of materials within the myelin sheath. Okay, so you can learn this spelling and practice it if you want to get it right. I think it's a German name. So we spoke about the process of myelination and we spoke about clefts within the myelin sheath. But can you tell me what is the function of the myelin sheath as such? You can type it in the chat. What is the function of the myelin? Yes, most of you are talking about, yes, gives insulation to the neurons. It has a role in nerve regeneration. And this insulation to the axon, it actually helps with a process called saltatory conduction of nerve impulse. Yes, the myelin sheath will help the impulse to travel from one node to another, and that is called saltatory conduction. Good. So you mentioned insulation, saltatory conduction. Then you can talk about protection and aiding in nerve regeneration. Those are the functions of the myelin sheath. Okay, so the stain for myelin, the special stain is called osmic acid. And you can see it is a black color stain. So here, this is the axon. The black color on either side of the axon is the myelin sheath. And the cleft within the myelin sheath is the Schmidt-Lantermann cleft. This is another special stain. Okay, it is fixed with osmic acid, but stained with toluidine blue. So here, this is a longitudinal section of the neuron. A is the axon. Okay. Then this region is the node of Ranvier. This thickened portion is the myelin sheath. So you can see within the myelin sheath, there are clefts here. Okay, this is the Schmidt-Lantermann cleft, the oblique cytoplasmic tunnel. This is the diagram. At the end of all this, this is the diagram that you're going to draw for the nerve fiber. Okay, so nerve fiber refers to the axon. So you're going to draw the central axon. Axon is also called the axis cylinder. Okay, axis cylinder is another name for axon. The cytoplasm of the axon is called axoplasm. The cell membrane of axon is called axolemma. So this arrow should actually be pointing at the cell membrane of axon. Now around the axon, okay, because the stain is osmic acid stain, you will be drawing it with your lead pencil. Okay, so please take note of these colors. In lead pencil, you will be drawing, see, this is one segment of the myelin sheath, and this clear portion is the cytoplasm of Schwann cell. And here you're going to draw the flattened nucleus of Schwann cell in an eccentric position. And from the Schwann cell, here there's a cytoplasmic projection. Okay, these projections you're to label as the Schmidt-Lantermann cleft. And in between the two segments of the myelin sheath, this region here is the node of Ranvier. This is how you will draw and label the nerve fiber in your record. This is just some extra information. You will be asked about the types of neurons. Okay, so as you can see, all the anatomy that we learned so far is actually the anatomy of the multipolar neuron, the cell body, dendrites, and axon. Whereas a neuron, which is called the bipolar neuron, there will be the cell body, a single dendrite, which will then branch into multiple processes. And the axon is, as usual, a long axon with branching at the terminal region. So there are basically two long processes emerging from the neuron. So it is called the bipolar neuron. Do you know the site where the bipolar neuron is found in the body? Okay. Tell me the site of multipolar neuron and the site of bipolar neuron. You can type it in the chat. Yes, bipolar neuron is present in the retina. Good. What about multipolar neuron? Where is it present? Yes, cerebral cortex. Multipolar neuron is also present in the spinal cord. Yes, good, good. And let's look at the unipolar neuron. Okay, also called this particular subtype of unipolar neuron is also called pseudo-unipolar. Okay, because here there is one process emerging, a short process emerging from the neuron. And this short process will further branch to form axon and dendrites. Okay, so you can learn this detail from your textbook. It is sometimes asked for you. And the site of pseudo-unipolar neuron, you already told me it is in the dorsal root ganglion. So classification of neurons based on the number of processes, you can classify as multipolar, bipolar, and pseudo-unipolar. Based on the function, we will be classifying it as motor and sensory neurons. Then based upon the speed of conduction of impulse and based upon the thickness of the nerve fiber, you will be further classifying it as Golgi type 1 and Golgi type 2 neurons. Okay, this classification of neurons, I think it's dealt with in detail in physiology when you're learning about nerve impulse conduction. So you can just go back and read that from your textbook in detail. Now we come to the last part of the class. Okay, there's one more slide for you to learn. Okay, we're going to learn about microscopy of the peripheral nerve. It is quite simple when we compare it with the microscopy of the skeletal muscle. So can anyone of you tell me, okay, you can raise your hand and then you can answer on the microphone, okay? Just describe the connective tissue of the skeletal muscle. Okay, it's already given in this diagram. Anyone wants to answer, you can just raise your hand and answer on the mic. Okay, tell me about the organization of the connective tissue in skeletal muscle microscopy, what you've already drawn in your diagram. You've already drawn it in your records. Yes, anyone with the answer? Something about what is endomysium, what is perimysium, and what is epimysium? I think you need to revise this topic, okay, about skeletal muscle. Yes. Anyone else with the answer? If anyone has the answer, you can tell it now.
Ma'am,
Yes. Yes.
Epimysium is covering the whole muscle,
Yes.
and perimysium covering the muscle bundles,
Okay. Good.
and endomysium covering a single muscle cell, muscle fiber.
Yes. Very good. Very good, Fatimatu. Good. Thank you.
Thank you.
Yes. So you can see in this diagram, you see, suppose this is a muscle fiber or a muscle cell, a single muscle fiber. It is wrapped with a connective tissue known as endomysium across around a single muscle fiber or a single muscle cell. Whereas many muscle fibers together will be forming something called a muscle fascicle or a muscle bundle. The muscle fascicle is surrounded by perimysium, whereas many muscle bundles or fascicles together will be ensheathed with connective tissue called the epimysium. Okay, so you need to know the difference between these three types. Muscle fiber surrounded by endomysium, muscle fascicle surrounded by perimysium, and the entire muscle, okay, a collection of fascicles surrounded by epimysium. So why I'm revising this is because the nerve also has a similar arrangement. Okay, we learned about the axon being the nerve fiber. It's these small dots here is the axon. Okay, the axon will be surrounded by a covering of connective tissue, which is called endoneurium. A whole group of axons will form a fascicle here. Okay, it's fascicle of nerve fibers, which will be covered with perineurium, and many fascicles of nerve fibers together will be enveloped with connective tissue, which is called epineurium. Okay, so you need to learn these differences. Endoneurium around a nerve fiber, perineurium around a nerve fascicle, okay, or a bundle of nerve fibers, and epineurium around a whole collection of fascicles. So this epineurium is actually covering the entire nerve as such. The entire nerve we see in gross anatomy is covered with the epineurium. Okay, this is what we spoke about. This is very important. It's a must-know topic for each of you. So this is the cross section of the peripheral nerve. This is a special stain, it's toluidine blue. So I'll just tell you that this is the axon. Okay, this is an axon. And if you see this whole group of axons, this is called a nerve bundle or fascicle. And this is the covering of the entire nerve. So based on this, I have one more question for you. Can any one of you just label this diagram? Okay. I've labeled the connective tissue sheath 1, 2, and 3. So can you tell me what is 1, 2, and 3? What are the labels? Yes, anyone can answer on the mic. Okay, I told you this is the axon. So 1 will be the covering of axon. Yes. Anyone with the answer?
One is endoneurium.
Yes. Good.
Two is perineurium.
Yes.
And three is epineurium.
Okay. Good. Good. Very good. Thank you for the answer. Yes. So you know that 1 is endoneurium surrounding the axon. 2 is the perineurium. This is a whole nerve fascicle. So this is 2, the perineurium, and 3 is the epineurium surrounding the whole nerve. So when we are drawing the histology of peripheral nerve, we'll actually be drawing the cross section of the thickest nerve in the body. Okay, so what is the largest or the thickest nerve in the body? You can type the answer in the chat. We had learned. Yes, good. The thickest or the largest nerve in the body is the sciatic nerve. So we're going to be drawing cross section of the sciatic nerve. Yes, so in this diagram, it will be similar to the diagram that you drew for cross section of skeletal muscle. Since the nerve is a thick nerve, okay, you will be drawing only part of the nerve within the diagram. So you draw it, okay, as an arc, and this is the epineurium. Within the nerve, you will be drawing fascicles of nerves. These are the nerve fascicles or nerve bundles. The fascicle is surrounded by perineurium. Within the nerve bundle, you can see, okay, this dot is the axon, and surrounding the axon, the smaller circle that you draw is the endoneurium. Okay, so you're drawing the dot axon surrounded by endoneurium, nerve bundle surrounded by perineurium, and the entire nerve fiber surrounded by epineurium. Now, in between the endoneurium, okay, in between the nerve cells, the nerve fiber, you'll be drawing a few flattened nuclei with hematoxylin. These flattened nuclei are the nuclei of Schwann cells. And within the perineurium, you'll draw a few blood vessels, a small blood vessel in the perineurium. And you'll also draw a few scattered fibroblast nuclei. These fibroblasts are what helps to create the connective tissue within the perineurium. So this is how you will draw the diagram cross section of peripheral nerve. Okay, this is just a few topics for extra reading, as I have mentioned. So all of you should know how to draw these 3 diagrams. Remember that for nerve cell, you will be using toluidine blue. So you draw it with your blue color pencil. For nerve fiber or for axon, it's with osmic acid staining. So you will be drawing it with your lead pencil. Whereas cross section of the peripheral nerve or sciatic nerve, you will be drawing with your hematoxylin and eosin colors. Okay, so thank you, class, for your patient listening. You have 3 more slides to draw that we'll be dealing with on Monday. So please draw the diagrams over the weekend in your histology record and go through this topic. If you have any doubts, you can approach me through WhatsApp. Yes. Thank you, class.
Thank you.
Thank you, ma'am.
Thank you.